So first of all, thanks a lot to Derek for organizing this amazing event. It's been a real pleasure so far, and thanks for inviting me. So as Jan also just talked about, there are several reasons why we try to do modeling. One of the best reasons in my mind is that while we know that the diffusion signal is extremely sensitive to physiological changes, we also know that its sensitivity is it's very sensitive, but its specificity is not so high. So the goal is that by modeling the signal and its connection to microstructural parameters, we can remedy this problem. It can also help us to, of course, design better sequences that better probe the things that we're interested in and so forth. There are several reasons that we do this. However, when we look at microscopic images of tissue, we become a little bit depressed, perhaps because it's extremely complicated, extremely complex. Perhaps one could be tempted to give up, that we will never be able to capture these things. And indeed I don't think we'll ever be able to capture everything. But luckily, I don't think we have to, because modeling is all about identifying the relevant degrees of freedom, so the features that actually affect the signal. So this is completely analogous to physics, where we don't need to take into account nucleons and quarks and gluons and so on to understand chemistry or to understand hydrodynamics. So the art is identifying which picture of microstructure that's the relevant picture to have, and ignoring all the degrees of freedom that we don't need. So once we have a good model, of course, we need to examine it very carefully. We need to try to validate it. And there are several ways of going about with this, and I will touch upon that later. And this is, by the way, unlike more mathematical descriptions of the diffusion signal, something you might call representations. They are still extremely useful, very sensitive, nothing bad about them. They are, however, unspecific. And examples of those are DTI and DKI. So in this picture, DTI and DKI are not models. They are representations, and I'll argue for that in a little while. So this model, the Neurite diffusion model, was developed during a research stay at St. Louis, Washington University, with Dmitry Yablonsky and Alex Substansky and Chris Kroenke. And the idea was that one of the characteristic features you see when you look at microscopic images of neurons are, of course, the dendrites and the axons, so collectively known as the neurites. So we thought that perhaps with first approximation, it could be an idea to try describing diffusion in such complicated tissues in terms of two non-exchanging compartments. So one compartment which describes diffusion in the extracellular space and in glial cells and so on, and one compartment which describes diffusion in axons and dendrites and the neurites. So the neurites, they look locally very much like cylinders. So cylinders are one of the geometries that we can actually describe with the propagator exactly. So that's what we're going to try to do with those, with the diffusion signal from those parts. So that means the neurites are individually approximated as long hollow cylinders with a longitudinal diffusivity dl and a transverse diffusivity which is approximately 0 because of the small size of this. So that, of course, that depends on the gradient, just to connect it with the last talk. And now I just start by presenting the model. I'll argue a bit for its validity regime later. So the actual collection of dendrites and axons within a voxel is then captured by the fiber orientation distribution function. So you can imagine, for example, here's a digitally reconstructed neuron in 3D, where you can try to estimate the distribution of these cylindrical segments, the neurites. So you get a distribution on the sphere, and you can represent it in many ways. You can expand it in spherical harmonics. That's kind of an obvious thing to do. You can also use parameterized distributions such as the Watson distribution and other more general distributions. They have each their advantages and disadvantages. So to recap, the Neurite diffusion model has two compartments, one which is internal to neurites, and basically the other one is everything else. And the neuronal population, or the Neurite population, is characterized by an orientation distribution function, an f of n hat here. And in the extra neuritic space, so to speak, we have Gaussian distribution. So an example of how a signal could look like would be like this. So we have an overall signal amplitude here. We have a Neurite volume fraction, nu here. And then we have a convolution over the fiber orientation distribution function with the kernel, so the signal from each individual Neurite here. And then we have plus the signal from the rest, basically. And here we have a diffusion tensor for that term. And you could also imagine including this, this part within the convolution itself, which is arguably more appropriate for white matter, for example. So the features that you get out of this is the compartment fraction, so that specifically the Neurite density nu. And then you get the compartment diffusivities, dl and d, so that the tensor really. And you have also the FODF, from which you can get scalar measures, for example, fiber dispersion, and what else? So to study the regime of validity of this model, it's very useful to go back to the cumulant expansion, which we also heard about yesterday. So generally, the diffusion signal is this average over the spin phases. That's very general. And this kind of average is very well known from probability theory. You can make an expansion in powers of the moments, basically. An expansion of log to this. It's known as the characteristic function. And then you just get this infinite series. So this is very, very general. And it's, in principle, it's not, it almost always applies. And it just amounts to saying that locally, you know, any curve, any reasonably behaved curve looks, if you look close enough, to some point it looks like a line. And if you look a little bit further, you may need to include some curvature and so on and so forth. So that's why I say there's not really any assumptions here. It's not a model. It doesn't hinge on any specific picture of microstructure. So written in terms of, so this is without flow and things like that, of course. So written in terms of B tensors and diffusion tensors, we get the familiar diffusion tensor part here and the ketosis tensor part here. And we get higher order terms as well. In general, we have an infinite number of terms. But for a fixed diffusion time, it's also clear from this kind of picture that we can always, for a given level of SNR, we can always select some, go down sufficiently long in B value and find a regime in which the difference between DTI and the signal is on the level of the noise. If we want to go a bit further, we need to include the next term and so on. So if you analyze this, you find that generally if the diffusion wave vector is not too large, then it's a good approximation to describe the Neurites in terms of Gaussian. So this doesn't actually mean technically that you say that diffusion is Gaussian. It just means that you work in the Gaussian regime. It's still restricted diffusion, but the propagator is actually described by a Gaussian propagator. So for our experiments where we have very, very high gradients, we go up to B values of about 15,000 typically in the animal scanners. We are okay I mean, we have very high Q values, so the highest Q value is actually below this, and that means that we are fine as long as we don't try to look at axons that are, you know, on the level of this 1.6 micrometers. So anyway, I would say this part of the theory is not so fundamental. You could always add ketosis, so intracompartmental ketosis terms to the kernel, so to speak, and add that in the model, so that can be repaired and become more accurate. What's probably more important is this assumption of no exchange, and I agree that this has not been established at all. Of course, there is exchange at some time scale, but what's important here is that there's not a lot of exchange during the diffusion time of our experiments. So here are some indirect measurements of the lifetime of water molecules inside cells. And they range pretty widely, so that underscores that it's not really known what it is. But all of them are at least higher than the diffusion times that we typically use of about 10 milliseconds. Okay, so I'm a bit in a hurry now, I can see. So we've done some histology to compare to myelin staining. shows both qualitatively with the neurite density, it fits pretty well. We did some correlation analysis and also got a pretty good agreement with the myelin stain. We did electron microscopy stereology and also found a good stain with the axon density. The dendrites the dendrite agreement was not as good, but if we look at the orientations of dendrites, for example, in the hippocampus, so here there's no myelin in these layers, we can still see that the FODF is captured quite well up here. This is from DTI, this is from the model. In those regions, it correctly captures the orientation of these. It's the same in the cortex, as we see here. We get these radial patterns from the model, which are also known from histology. Now, like I said, the diffusion tensor is quite general. So if you have a model, you can also compute the diffusion tensor. And here's the example from the Neurite diffusion model, where you can see it predicts that the anisotropy of the measured diffusion tensor is caused exclusively in this model by neurites. So it's weighted by the neurite volume fraction, but only if the neurites have anisotropic diffusion themselves, and only if they are oriented in an anisotropic pattern. These contributions, by the way, can be separated. I mean, these internal contributions and this orientational contribution can be separated with the double diffusion encoding, like this. So we tested this by simulation and digitally reconstructed neurons using Monte Carlo simulations. We found a very nice agreement with this. Oops. These are just the centralized eigenvalues. The eigenvalues minus the mean of the 3 eigenvalues, so that's why it's negative. So we got a very nice linear agreement between the eigenvalues of this scatter matrix. I didn't say what that was, the T. So that's basically the second moment of the orientation distribution function for the neurite patterns, which could be measured from these digitally reconstructed neurons. You can also use animal models where there's neural plasticity to test whether the model can actually capture these changes in the neurite architecture. So here's a model of normal development, where for the immature cortex, the dendrites are very sort of simple, one-dimensional more or less. And in the more mature cortex, collaterals like this start to develop. So we did on a ferret brain, on an immature ferret brain, we did histology. So we tried to measure actually with histology these FODF, so the orientation patterns of the dendrites and axons from different places along the cortex, like this. This is a picture from before. And then we compared it with the DTI using this model. So here this is first a model independent comparison. So we plot the FODF here. I just want to put a focus on the last 2 columns here. So for this part here is the FODF as determined from histology. And this part here is basically the ADC as a function of direction. And no independent co-registration has been performed here. So we just see that that the FODF actually, the diffusion ODF actually captures the correct peak position of the neurites obtained from histology in all cases. It's much wider, of course, but that's because of the cosine squared basically behavior you get from the ADC. And it also happens that the distributions over here that are more narrow are also more narrow here than in the other 2 cases down here. So that looked pretty good. Here's the main eigen vector from diffusion tensor. That's the yellow lines compared with the main direction of the histologically determined direction with the red lines here. And we see in general there's a good agreement. 2 slices shown, and quantitatively also we tested whether the angle between them was significantly correlated or not. And we see here that it's a pile up around 1 here, very significant, indicating that there's a high degree of alignment between those 2 as the theory predicted. So by plotting the eigenvalues from the diffusion tensor and the eigenvalues from this scatter matrix determined by histology, you can test this linear relationship that was predicted, and you can determine the slope, which gives you this product of neurite density and diffusivity. And then you need additional information to separate those 2. And here's an example from, I believe that was from an NAA study using ratios of free water to measure NAA diffusivity, and assuming the same for water, you could get an estimate for neurite density in this way that fits very well with literature values. So we've recently generalized Matt Boddy's structure tensor approach. He developed it for 2D as a method to automatically capture or estimate directions of neurites in tissue. And we extended that to 3D, shown an image here. So that will in the future enable us to do more larger scale comparisons of these things. So of course you can continue this, so not only the diffusion tensor, you can also go to the ketosis tensor and link that to the microstructural parameters. And the advantages of that is that you perhaps don't need as much data to estimate diffusion tensor and ketosis tensors as you do to the full Neurite diffusion model. The data that we have, because of the many parameters, we need really to acquire a lot of data. It takes a long time. And we need to, of course, to test for local minima and so on. But this relation looks quite complicated, and it's not very easy to sort of invert that and get these internal model parameters from the measured ketosis tensor. This is just the mean ketosis tensor. You have all the mean of the ketosis tensor. You have all the other elements of the ketosis tensor as well, of course. However, if you go to very simple regions, so for example, spinal cord, where the axons and the dendrites are highly aligned, these relations simplify significantly, and it's actually possible to do this inversion, so to estimate the intra-axonal diffusivities from the ketosis tensor and the diffusion tensor. And we were able to do that with Norm Shemesh in combination with this recently developed fast DKI, so a method where instead of acquiring 60 directions, as I think is typical, we've shown that you can make do with 9 directions on 2 shells and get a really accurate measurement of the most important ketosis parameters, so the mean ketosis, the radial and axial ketosis. And then you can use that saved time to acquire measurements over a large extent of diffusion times here from, I think, around 3 to 350 milliseconds of diffusion time. So inverting those relations, we find this is what I called DL before. Sorry about that. We can actually get time dependence of according to the model, intra-axonal diffusivity over this large time range, and also of extracellular diffusivity, perpendicular and parallel components here to the spinal cord. And what you see is that, for example, down here for short diffusion times, they are almost equal, these two, so you approach a kind of a free diffusion limit. And as time goes longer, this the perpendicular one goes to 0 or becomes smaller. So nevertheless, even due to these accelerations, it's still a very time-consuming technique. So we several years ago we looked at some ways to try to speed this up by averaging over directions, a so-called powder average, and we found some good results for the neurite density, but the other parameters were problematic. They were not estimated very well. And then others have been more successful with a similar kind of approach. And I think recently the New York group has extended it even further, not just by the sort of zeroth powder averaging, but higher order spherical harmonics as well. Okay, so I don't have time to talk about this. So we have some stress models also that show the expected changes. I just want to mention this really nice recent work, which I was not involved with, by Ferrat, Firmans, and Novikov, where they did a different kind of validation, so inspired by physics kind of view, where, for example, in physics you don't have any histology, right? So how do you validate? How do you prove theories? And the typical way is to drive experiments out in a range where they have very characteristic functional dependencies. For example, for this Neurite model and its in similar models, you can show that for very, very large p-values, only the error function term of the powder average signal survives, which in turn, and there's an error here, so I should point out, I was very disappointed I just discovered that. This should be 1 over square root of BDL here, for very large values of BDL. And what they did was they isotropically averaged the signal over 64 directions from 0 to 10 milliseconds per square micrometers. And then they replotted this signal as a function of 1 over square root of B. And if this theory is correct, then it should behave, all the points should fall on a straight line, which it looks like it does very, very nicely here. And you can even add the old data from Chris Cronke's NAA paper back from 2004, rescaling for the difference in diffusivities, and you find that that also falls in that line. So I would say that this, for the gradient strengths used here, this is a very good evidence, I think, for this zero radius picture, because otherwise we would not have this 1 over square root of BD dependence. If there was a finite radius, it would decay exponentially. So I have a lot of collaborators on these different things that I would like to thank, and some funding as well. So thanks very much.
Okay.